Hello everybody and welcome to Mr. Saucedo's YouTube videos. Today we're going to be talking about exceptions to Mendelian genetics. So we've spent this entire time talking about monohybrid and dihybrid types of situations. And so the thing about Mendelian genetics are that there are obviously exceptions to that. They're not things that sometimes are purely dominant or purely recessive. And so we're going to talk about the four major exceptions to Mendelian genetics. So it's kind of lucky that Mendel, when he was looking at pea plants, the traits that he was looking for were purely dominant or purely recessive, because there are lots of situations where that isn't the case. So we're going to talk about one of those first, which is called incomplete dominance. So incomplete dominance occurs because some alleles are sometimes neither dominant nor recessive. And so what that means is that they actually blend together and create a characteristic that is different from both parents. And so you can think of it, a good example of that would be like red snapdragons and white snapdragons. So if you crossbreed pure red snapdragons with pure white snapdragons, you actually get an F1 generation that is all pink. And so you can put that down as a pretty good example of like red times white equals pink, because that's a good example of incomplete dominance. And so a good human example of that would be hair texture. So it's actually more of a spectrum for hair texture. And so even though we like to, when we're doing Punnett squares, we like to say, okay, well, curly, that's dominant and straight is recessive. It's actually more incompletely dominant, which means that it's possible that you have, you know, um, incomplete dominance and end up having wavy hair as a result of crossing a curly haired parent with a straight haired parent. You could end up getting wavy hair instead, and that's incomplete dominance. So again, in a Punnett square, this is kind of what we're talking about. If you had, two pink parents, you could end up with, 25% of their offspring being red flowered, 25% being white flowered, and then about 50% looking exactly like the parent and being pink. So here's what we're going to do. We're going to do an example of incompletely dominant Punnett square stuff. And we're going to kind of simplify things a little bit. But the idea is almost everything is the same. The difference, though, is the way that we write dominant and recessive, right? We're used to capital letter being dominant, lowercase letter being recessive. Well, for incomplete dominance, we use capital letters and we use superscripts to distinguish between the different alleles. So like this could be like the white colored flower, and this could be like the red colored flower. So it's the superscript here, the little letter that we write like an exponent. That is what's distinguishing between them. So let's say that we have this crossing. So let me explain. So CB, we're going to say that's like a blue flower. CR, we're going to say that's like red. Okay, that's the allele for red. That's the allele for blue. So blue and red, if that's incompletely dominant, that would be a purple flower. So we're looking at a purple flower and we are looking at a red flower, and we're going to be crossing them. Okay, so we still do the exact same thing. We separate our alleles. We go column by column, and then we go row by row, fill them in. So this would be the result. And so genotypically, 50% of them are homogeneous. Sorry, I almost said homogeneous. Are homozygous for red. 50% are heterozygous. That means that 50% of them would be red. 50% of them would be purple. And that's incomplete dominance. Okay. We also have situations called codominant genetic crosses. So codominance occurs when two alleles are dominant, and so both traits get expressed completely at the same time. So instead of a mixture, so instead of a blending like incomplete dominance was, we're looking at a mixture when we're talking about codominance. And so if you have like a red bull, not the drink, and you have a white cow, and they reproduce, if, we're going to call it like hair color, right? I guess it'd be like hide color. I'm not really sure what you would call it, but we're looking at the color here. You could end up with like a white, um, a white bull with red spots. So we end up with like, like a mixture of both traits. And so this is called a roan um, bull. And so a good example of that are freckles. So freckles are codominant, meaning that you end up with like a mixture of traits that are in charge of freckles. And so here we go. Red times white equals white with red spots. That's a good example. Again, an animal example. I thought that this was a great example also of like how to make pandas. So if this were true, that's why pandas would look like this. It's not true, obviously, but this is a good example of codominance. So instead of just having one or a blending of the a blending, so if this were incompletely dominant, all four offspring would more likely than not be gray, which is like kind of a blending of the two colors. But instead, we have little bits that are have little black parts and white parts, and so that would be an example of like codominance. So for codominance, it's the exact same. We still use superscripts because dominance and recessiveness and all of that stuff doesn't really make a lot of sense when we're talking about codominance. So let's do this. We're going to look at like white and red. Okay. So we have two parents that have a mixture of white and red, let's say spots or something. Okay. They're codominant. So we would still split them up. We would still go column by column and then row by row. And what you would end up with is 25% are homozygous for just red. That's it. 25%, or sorry, 50% are heterozygous. 25% are homozygous for white. Now, if we were looking at them though, what would they look like? Well, we would have one that would have just the white traits. So that would mean 25%. We would have 50% that would have both traits, so like white with red spots or red with white spots. And then we would have one that would have only the red traits. And that would be an example, again, of codominance. So here's a good one. We have camellias, white camellias and red camellias. This is what their child ended up looking like. Is this an example of codominance or incomplete? Answer, it is codominance. We can see both traits being expressed. Here we go. We have some bell peppers. Answer, incomplete dominance. Red and yellow makes kind of an orange color, and it is definitely a blending of the two. We have a black chicken and a white rooster. They mate and they have the following offspring. That's an example of codominance. Here we go again. We have some mice. That would be an example of incomplete dominance. Black horse and white horse have the following offspring. That would be codominance. All right, what about multiple alleles? So far we've pretty much had dominant or recessive, right? So that's it. There are only two alleles that are possible. And then we now know that we can blend or we can mix. So that's not really a different allele, though. That's really just, well, you guessed it, right? It's just the expression that is a little bit different of those. So multiple alleles, though, some traits do have more than two. The most common example is blood type. So for blood type, we have a whole bunch of different possibilities. And so we have O, A, or B, and you can have any mixture of those. You can have O and A, we just call that A. We have O and B, we just call that B. You have A and B, we call that AB. So there are plenty of examples of multiple alleles where if one parent has A, B, or O, and then another parent has A, B, or O, these are kind of like this is what would be mixed together if we were looking at like an actual chart. So we're going to talk about multiple alleles, and normally you need like a quick chart like that on the previous slide in order to figure out what's going on. But here is an example of blood type. So this parent has A type blood because O is nothing, basically. O has no markers. So whatever, if you have no markers, right, you're O. But anything else like A or B or AB, those are expressed. So this person, this parent would be A. This parent would be AB. Okay? So we still split up everything like this. We still go through column by column and row by row. And so what exactly is the breakdown for this? So again, you wouldn't really know unless they gave you a chart. So here is our chart. If you have this, you're O type. If you have this or this, you're A. This or this is B. This is AB. Okay, so now we can interpret this genotypically. So 25% are AA, 25% are AO, 25% AB, 25% BO. They're all different. But phenotypically, what type blood do they all have? All right, well, 50% have A-type blood because there are two ways of making A-type blood, and we have those two ways right here that are present. 25% would be AB, 25% would be B. And again, you would need this in order to figure that out. We don't assume you know the mixing for blood type. Okay. Last but not least, and we're not going to do an example of this really for Punnett square stuff, but polygenic traits. Most traits that we are talking about are actually controlled by many different genes, sometimes on multiple chromosomes. And so the most complicated traits that we have are normally controlled by several different genes. Eye color, skin color, hair color, all of those are controlled by a selection of multiple genes. And so doing a simple Punnett square isn't necessarily the easiest way of predicting those types of things. We do like to simplify, but it's not exactly as simple as we like to make it be. All right, and that's it. So if you have any questions, let me know.